The mesolimbic pathway is also known as the reward pathway in the brain. It originates in the ventral tegmental area, or the VTA, and stretches to the nucleus accumbens, predominantly through dopaminergic neurons. When activated, the VTA releases dopamine via exocytosis into the synapse where dopaminergic neurons transport the dopamine to the nucleus accumbens and the amygdala. The nucleus accumbens plays a role in reward and desire, while the amygdala is the anatomical structure associated with emotion. Thus, both working concurrently is said to play into conditions such as addiction and depression. Opioid addiction, specifically, abuses this pathway through inhibition of calcium release, inhibitory effects of serotonin on dopaminergic neurons, and/or the stimulating effects of norepinephrine. Opioid drugs fall under the category of narcotic analgesics. This category contains drugs that are very successful in reducing pain without causing unconsciousness. They are also known to create a state of relaxation and sleepiness, and have some strong potential for abuse and overdose. Opium specifically comes from the poppy plant and is a source of the family of drugs called opiates, aka opioids. Opium is prepared by gathering the milky juice from the poppy plant's pre-ripened seed pod, and then is dried and powdered. Opium contains codeine, thebaine, narcotine, and others. However, the principal active ingredient is morphine. The discovery of morphine was revolutionary. It's the first of its kind to be isolated and allow painkillers to be administered in known dosages. Opium has many different derivatives that produce a variety of effects, one of which is codeine. The opiate codeine is an effective cough suppressant and nearly identical structure to morphine. However, its differences result in milder analgesic effects and less side effects in general. The morphine molecule became extremely helpful when modifications were found to generate other drugs that had variations in oral effectiveness, potency, and other action duration.
Beta-endorphin is an opioid peptide that also binds to mu receptors. When beta-endorphin binds to a mu receptor on the nerve terminal of a GABAergic neuron in the ventral tegmental area, it inhibits calcium channels. This reduces the chances of reaching an action potential and thus decreases the firing rate of GABAergic neurons, which then release less GABA onto dopaminergic neurons. So overall, beta-endorphin also increases the rate of firing of dopaminergic neurons and increases the release of dopamine.
Primary nociceptive afferents in the superior cochleus have mu receptors on their terminals. Spinal opioid interneurons can then release endorphins in this area, which will bind to mu receptors. This binding inhibits the release of calcium from the nociceptive afferent, reducing its ability to release glutamate and substance SP from the afferent neuron. SP is normally co-released with glutamate in response to painful stimuli. Dorsal horn neurons can also be inhibited by mu receptors and opioid binding. These spinal projection neurons express the mu receptor. When the opioid interneurons release endorphins onto the spinal projection neuron, this activates their potassium channels, inhibiting their action.
Mu receptors are represented in the periaqueductal gray on nerve terminals of neurons. Opioid action in the PAG is reliant upon the interactions of GABA interneurons. When opioids bind to mu receptors located on GABAergic interneurons, they inhibit calcium channels and reduce the firing of these neurons, and decrease the release of the inhibitory neurotransmitter GABA. This increases the release of serotonin into the PAG because GABA's inhibitory control over serotonin neuronal firing has decreased. Mu receptors are represented in Rafe nuclei and in the locus ceruleus on nerve terminals. The LC is an important area for the synthesis of norepinephrine. Normally, adenylyl cyclase inside of the cells in the LC convert adenosine triphosphate, or ATP, into cyclic adenosine monophosphate, or CAMP, which is what triggers the release of norepinephrine. When an opioid binds to a mu receptor on the terminal of a neuron in the LC, it inhibits the action of adenylyl cyclase through the same calcium mechanism. This decreases the amount of CAMP produced and thus decreases norepinephrine release. Behaviorally, this is related to feelings of drowsiness because the release of norepinephrine in the LC is normally linked to our body's abilities to modulate our sleep-wake cycles. The derivatives differ in their ability to perform in the same way as opium. Heroin and methadone, for example, are full agonists, meaning they are capable of producing the same maximum response as opium. There are also partial antagonists, which bind readily to opioid receptors. However, they are not as effective. Some examples include Nubain and Buprexnix. When administered alone, these drugs produce partial opioid effects. However, if administered with an opioid of higher efficacy, these two compete for receptors and the higher efficacy drug is less effective. Lastly, there are pure antagonists. These are drugs that have a similar structure to opioids, but they don't produce any pharmacological activities. These drugs have the ability to reverse the effects of opioids since they compete with the same receptors.
Drugs that fit these categories are naloxone and nalorphine. Naloxone binds to mu receptors, blocking the binding site and prohibiting an opioid from binding here in a competitive antagonist fashion. Naloxone produces no pharmacological activity and therefore no efficacy, and no psychoactive effects other than reversing opioid effects. This is why naloxone is approved by the FDA to use as a remedy in opioid overdose situations.